Did it not say one?
It says 101, yes, on that.
Oh, it did? Did it flyer?
And it was like the one that was sent a few days ago, and then I think there was another one that was sent. That
Jan
was
was
one.
Rose waiting somewhere else.
They'll find out after like 5 minutes of chemistry talk.
Yeah.
You ran straight in the other direction.
Yeah. He's looking for it.
Odd numbering system in this place.
All right, well, thanks everyone for coming to our special seminar time. I'm super excited to be able to introduce my colleague Manny Aranda from King Abdullah University of Science and Technology in Saudi Arabia. Manny got his PhD from the University of Cologne in 2006. So Manny is Spanish, but he's raised in Germany. So he presents to me as German, but he's actually Spanish. Anyway, degree from the University of Cologne. Actually his background was originally in Evo Devo work in evolution of gene regulatory networks in insects. He did his PhD on that project and continued on to a postdoc for 3 years. But then in 2009, he joined Chris Bolster's group at KAUST and started as a postdoc examining the effects of UV on coral larvae. And he joined the faculty there in 2012. And he has really grown to be really a world leader in the genetics, cell biology, and genomics of the study of coral symbiosis. What are the underlying regulatory mechanisms that drive the symbiosis? And more recently, his lab has become very interested in sort of solutions to reef decline, selective breeding, assisted evolution, trying to build resiliency and help corals do better in our changing environment. I met Manny in a blizzard in New Jersey. We were stuck in a conference center together at Rutgers in 2014, right? Yeah. Where the snow was so deep that the organizers could not get to the building to start the meeting. I don't know how they fed us. Somehow the people in the conference center must have slept there, but we were fine, but our organizers couldn't get there. So that was a rather formative introduction to Manny. And he was this unknown to me because, I've been around corals for a long time, and suddenly there's this young whippersnapper coming in and doing all this really cool stuff, all these amazing techniques. And he hasn't slowed down, and his lab really is a leader. He's had over 20 graduates. He couldn't even count them. And he's had 10 postdocs. So KAUST allows one to have a very large research program, and they're afforded a lot of really awesome resources, and Manny has not squandered that at all. So thank you so much for visiting Oregon and for visiting OSU. It's been a great couple of days. Welcome.
I thank you for this nice introduction and you for attending this seminar, giving me the chance to show you a bit about the work that we've been doing in the past years. This is a summary of our work on symbiosis that we started some 7-8 years ago. And it's multiple studies that I put together to give you an idea of how we've been developing our work to study this relationship that cnidarians have with these photosymbionts. But before I dive into the actual experiments, I would like to kind of set the frame by giving a bit of background information. I assume that most of you are aware of what corals are, but just to freshen up a little bit, the reason why I study corals is because they're not just charismatic, they're actually important ecosystem builders. They're the keystone species of one of the most productive and biodiverse ecosystems on our planet. coral reefs. And although they only cover a small portion of the ocean floor, they're immensely important for our marine biodiversity. They support more than 30% of all named marine species. But they're not just important from an ecological perspective, they're also economically important to us. They support fisheries, and in many parts of the tropical world, coastal communities rely on reefs as the main source of protein. There's also many commercial fish species that require reefs at one time in their life. And these ecosystems also provide sources of income through tourism. A hectare of touristically exploited reef is worth about $1.5 million every year. So it's a substantial amount of money that goes into the economies. But they also provide some services that are a bit more difficult to put a monetary value on them. Like, for example, shoreline protection, right? So they break the waves before they hit the shores, protecting our shorelines, which means that we don't have to build structures to do the job for us. They also provide other services. There's a lot of bioactive compounds that are built or produced by coral reef species that can be used for medicinal purposes. In total, it is estimated they could be contributing up to $9.9 trillion to the global economy every year. And more than 500 million people on this planet are directly or indirectly dependent on these ecosystems as main source of income. But unfortunately, these ecosystems have been in decline for the past decades. We have already lost about half of the coral reefs, and the remaining half is highly threatened. And the main threat has now become climate change. There's other threats like pollution, coastal development, but in the past years, climate change and increasing sea surface temperatures have become the main threat to corals. And to understand why this is the case, we first need to understand what a coral actually is. Some people believe they're rocks, but in fact, they're animals. And they're what we call metaorganisms. So they're organisms that are composed of multiple species that together build what we call a holobiont. Most prominent, of course, is the coral host in the middle, and a plethora of microorganisms like bacteria, archaea, fungi, and most importantly, what we call the photosymbionts, dinoflagellates in the family Symbiodiniaceae. Now, corals are basically these little polyps. Many species form colonies where you have hundreds and thousands of polyps living together in a colony. And their body plan is actually quite simple. They only have two tissue layers, an epidermis. This is also the tissue layer that carries the signature stinging cells, the cnidocytes or nematocytes, and a gastrodermis, which is an inner cell layer surrounding the gastrodermal cavity. And it is in this cell layer where we find the symbionts. They sit as endosymbionts within gastrodermal cells. So it is a very intimate symbiotic relationship where the symbiont is allowed to reside within the host cells. You can actually see quite nicely here in this picture, the red color is the autofluorescence of the chlorophyll from the symbionts. And you can see that the polyps are densely packed with symbionts. Now, for reasons that we don't fully understand, the relationship they have is kind of sensitive to environmental stress. For example, increased temperatures can lead to what we call coral bleaching. For unknown reasons, it is expected that it has to do with the breakdown of photosynthetic machinery of the symbionts. The host triggers the expulsion of the symbiont from its tissues. And now this thin tissue layer becomes transparent, and you can see the white calcium carbonate skeleton shine through that tissue. And this is why we call this, this response coral bleaching, because the colony turns eerie white. At this state, the coral is not dead, it still lives, but with its symbionts, it lost its main source of nutrition, of food. So in this state, it can survive only for a certain time. And if the temperature doesn't go back down and allows the coral to repopulate the tissue with symbionts, the coral basically dies and the skeleton gets overgrown by algae. Unfortunately, this has been happening on global scales. We now had several global mass bleaching events where we had corals and entire reefs around the world bleach. So it is a central question in the field if there's something we can do to prevent this from happening. But before we can really understand or think about how we can change this, we need to understand what is this relationship they actually have? How does it work? What is it that the host gets? What is it that the symbiont gets? How do they interact on the molecular level? Do they actually cooperate? And most importantly, if I allow somebody to live inside my own cells, how can I control his own proliferation, that he doesn't proliferate at my own expense, right? So these are very important questions that we wanted to answer when we started this work. And because corals are so complicated to work with, we actually made use of this wonderful model that was introduced by Virginia. So thank you very much for this. And this is the sea anemone Aptasia. Can you actually start the? Yes, you can. So this one is actually an aposymbiotic individual. And this is what makes it so nice to do this kind of research. In corals, because they're obligate symbiotic, we don't really have a non-symbiotic control for our experiments. We have nothing we can compare the symbiotic state to. But in this organism, we can actually keep them happy without symbionts as long as we give them food, they're happy. So we can now compare a symbiotic anemone to a non-symbiotic anemone. And this is exactly what we did, not only us. So we can take our anemone without symbionts, we can then give it the symbionts, and it's the same symbionts that we find in corals, to turn it into a model that resembles corals. At least we have an Idarian host and a Symbiodiniaceae partner. And we were not the only ones to do these experiments. There were multiple groups who were doing the exact same experiment, comparing gene expression profiles of the host between symbiotic and non-symbiotic anemone. And the nice thing was that all these three groups used exactly the same anemone genotype. So we basically performed the same experiment in parallel in different labs. And we found in total 4 data sets that were generated with this anemone genotype. So we downloaded the data and then analyzed it using exactly the same pipeline. And if you're interested in transcriptomics and in things like batch effects and experimental biases, I suggest you read a bit about this publication that we published in PLOS Genetics. For now, I just give you a summary. But one thing that you see immediately is, although we all use the same anemone genotype and did exactly the same comparison, the number of differentially expressed genes is vastly different between the experiments, right? So the lowest one found 2,398 differentially expressed genes, while the highest one found almost 12,000 differentially expressed genes. These are the 4 studies. These are the initial of the first authors. These are basically then the number of differentially expressed genes, how many were upregulated, how many were downregulated. So the first idea we had is, well, let's see which genes are differentially expressed across all these experiments. And what we found is that only 393 genes were differentially expressed in all 4 experiments, although they did exactly the same experiment, right? So thousands of differentially expressed genes, but only 393 were consistent across these 4 experiments. However, if you look at the number of upregulated and downregulated genes that were in common, you sum them up to only 300. Meaning that of these 393, there were 93 genes that, yes, were differentially expressed in all experiments, but in some, they were going differentially up, and in others, they were differentially down. So we end up with only 300 genes in common. So instead, what we thought is, what if we integrate all the data and we run a meta-analysis? And that's basically what we did. And with this, we identify 731 genes that we believe were high confidence symbiosis core genes that responded in all of these experiments. Now, because you cannot just look at 731 genes and try to make sense of it, we did a high-level analysis looking for enrichment in certain biological processes or INCAC pathway maps. So we try to find out, do we find clustering of these genes in certain biological processes? And what is interesting, it's maybe hard to read for you, but what we found was many, many processes where we have amino acid biosyntheses being enriched in these 731 genes. And when we looked at the CAC pathway maps, we found basically the same thing. Tyrosine metabolism, glycine, serine, threonine metabolism, biosynthesis of amino acids, and very important for the rest of the talk, nitrogen metabolism. So we looked specifically at these amino acid biosynthesis pathways and tried to make sense out of them. And what we came up with is this interesting switch in a pathway to produce the amino acids glycine and serine. And I'm trying to walk you through this. So what you see here are basically the two pathways that you can use to produce glycine or serine. In blue, you see the gene expression of the enzymes. And blue means that these are downregulated in symbiotic anemones. And in red, we see the genes that are upregulated in symbiotic anemones. And what this pathway basically translates to is that non-symbiotic aptasia would take up choline from food, for example, convert it to betaine, sarcosine, and then to glycine and serine. So it was used food to produce amino acids. However, when they become symbiotic, they downregulate these genes and instead start upregulating these genes here, which is a glucose transporter, and then enzymes that produce 3-phosphohydroxypyruvate as a carbon backbone. And then the GS-GOGAT machinery that allows to assimilate ammonium to that carbonate backbone to produce O-phosphoserine and then serine. And then serine and glycine can be converted into other amino acids like cysteine, for example. Now, because this was all based on gene expression, we asked, is this actually happening? So we repeated the experiment, but now we labeled the anemones with 13C bicarbonate, which the anemone itself cannot take up, but the photosymbiont can convert it to CO2 and use it for photosynthesis to produce 13C labeled glucose. And then we extracted the metabolites and looked specifically at these intermediate metabolites in the pathways to ask, do these intermediate metabolites respond like we would expect it from the gene expression profiles we found? And if the metabolite went down, we indicated it with a blue minus. And if the metabolite was enriched, we indicated with a red plus. And what you can see is that the metabolome analysis basically mimics what the gene expression profiles show. More importantly, we also could track that the glucose, so the carbohydrate backbones for these processes really came from the symbionts because we could find our 13C from the bicarbonate incorporated in there. Now, based on this, we then try to come up with a model how this could explain how the symbiosis actually works. So if we have a non-symbiotic anemone, we basically have food that we use to produce amino acids. Any waste nitrogen that is produced is converted to urea and excreted into the environment. However, once I have symbionts come in, at the very early stage, I have just a few symbiont cells that produce a negligible amount of photosynthesis. So we believe that these cells can actually use most of the glucose and all the waste nitrogen that is produced by the host to proliferate. So you have few cells come in that start to proliferate at a high rate, and this is what we see in the early stages of symbiosis. However, at one point, you start having more and more of these symbiont cells that start to compete for the nitrogen in the system. So they now start to limit themselves because they compete for the same nitrogen in the system. And then they reach the point where they start producing more glucose to photosynthesis that they can use for their proliferation because they don't have enough nitrogen to keep proliferating at the same pace. And that is basically when we reach a stable symbiotic relationship where the symbiont population now produces an excess of photosynthesis that it can translocate to the host. The host, however, can now use these carbon backbones to control the ammonium in the system by assimilating it himself into amino acids. So now basically I have a stable system where the translocated glucose from the symbionts is used to control how much ammonium is accessible for the symbionts to maintain a stable symbiont population. At least that was the theory. So the next question was, is host-dependent nitrogen recycling the mechanism through which the host controls its symbionts? Now, if this is really the case, it would mean that the symbionts must be nitrogen limited in hospity, right? Because the host uses this mechanism to control the nitrogen availability. So we went to the coral Psallophora pistillata, and while we cannot compare it to a non-symbiotic coral, what we can do is we can actually look at what is the symbiont doing. And the nice thing about Psallophora pistillata is that it associates with Symbiodinium microadriaticum, a strain that we can keep in culture. So we can actually have it free living under normal culture conditions. So we asked, what if we have these symbionts in culture and we start limiting the nutrients to see how the gene expression responses to that? And the first thing is we took the medium that we used and we partitioned it into 5 major nutrient groups: nitrogen, phosphate, iron, trace metals, and vitamins. And then we started experimenting by lowering each individual category to see, can we reduce proliferation rates in culture to about half? Okay. And what we found is that trace metals and vitamins don't matter. Even if you remove them completely, you do not get a 50% reduction in proliferation. However, if we reduce nitrogen to 1/4, phosphate to 1/8, or iron to 100th of the original concentration in the F2 media, we would get about 50% reduction in proliferation rates. So we said, okay, let's take this. So we subjected cultures to either nitrogen limitation, phosphate limitation, or iron limitation. And then we had our coral where we could extract the symbionts and the RNA to have this as another sample for in-hospity symbiont gene expression. So we had control, no nutrient limitations, nitrogen limited, phosphate limited, iron limited, and in-hospity in the coral Psallophora pistillata. And we did all of these RNA-seq comparisons. And this is the comparison of the gene expression profiles of the different nutrient-limited conditions to the in-hospity condition where the symbiont is in the coral. Here we see the log fold change of the genes in the iron-limited condition, and here in hospity condition. And if the gene expression profiles are very similar, what we would expect is basically a 45-degree line, showing that the gene expression changes is the same in both treatments. However, we see that there is almost no similarity between iron-limited cultures or symbionts in the coral, right? Their correlation, the Spearman correlation, is very, very low and insignificant. And the correlation between phosphate-limited cultures in in-hospity symbionts is also very weak. So very weak correlation of 0.02 and also not significant. However, when we compare the nitrogen-limited culture with the in-hospity symbionts, we now start to see a very strong correlation of the gene expression profiles and a correlation that is 0.83 and highly significant, suggesting that the symbiont gene expression profile in nitrogen-limited cultures is very similar to being in the coral host. And if we look at the single genes, this line here is basically sorted the fold changes of the differentially expressed genes in our nitrogen-limited culture. And because they're sorted by fold change, they give you this nice line. And then the dots represent the respective gene expression level fold change of the same gene in the coral host. And you see that the genes do change in the same direction, but in hospity, in the host, the gene expression difference is even more pronounced. So it looks like a nitrogen-limited culture, but all the gene expression changes are even more pronounced. So the final question to ask was, okay, this seems to indicate that yes, symbionts inside a coral host are nitrogen limited. So can we prove this experimentally? So we took a coral and we subjected it to different nutrient treatments. So a control, iron enrichment, phosphate enrichment, nitrate enriched, and ammonium enriched. In here, we have the symbiont density on the y-axis. And what we can see is that iron enrichment and phosphate enrichment did not significantly increase symbiont density. However, both nitrate and ammonium significantly increased the number of symbionts we found in the coral, which kind of proves that, yes, symbionts are nitrogen limited in Hospity because if we give them nitrogen, they do increase in density. Good. So the next question was, how is this recycling really happening? I mean, we already had an idea of this pathway, but we wanted to understand, we have a coral or an anemone, and we usually take the entire organism, mush it up, extract the RNA. But we already know that the symbionts are only in the gastrodermal tissue, right? So we're basically looking at a whole organism profile to understand something that is actually happened in discrete parts of the organism. So we thought, can we refine this actually further? And what we did is we used laser microdissection to separate symbiotic and non-symbiotic tissues. So what you do is you take frozen thin sections of your anemone and you put them on a membrane slide, and then with a laser microdissection microscope, you can basically cut out with the laser certain parts of the tissue and then collect it in a tube to extract RNA. And we did that. We did this with symbiotic and with non-symbiotic anemones, and we extracted the epidermis and the gastrodermis. So basically, we had 4 samples: aposymbiotic anemone epidermis, aposymbiotic anemone gastrodermis, and then the same thing, symbiotic epidermis and symbiotic gastrodermis. And then we looked at the gene expression profiles and classified the genes based on different modules. So either modules that respond to symbiosis, or genes that respond to aposymbiotic tissue, or genes that responded to gastrodermis specifically, or genes that basically respond to epidermis specifically. And when we then looked at the genes, and I'm only going to focus on the first module because this is the most interesting for this talk, the genes that are expressed in response to symbiosis, both in the non-symbiotic epidermis, so the epidermis of the symbiotic anemone, but it doesn't carry any symbionts because they're only in the gastrodermis, right? And the symbiotic gastrodermis, what we found was enrichment for genes involved in carbon and nitrogen cycling. And we already had kind of an idea what we would find when we look into this, and this is exactly what we saw. The genes of the GS-GO-GAT mechanism that I showed before and the TCA cycle. So basically the same pathway we identified before, where the sugar from the symbiont is used as carbohydrate backbone donor to assimilate the waste nitrogen. But the interesting thing here was that these genes are not only active in the tissue that carries the symbionts, but also in the adjacent tissue that does not itself have symbionts inside. So it is also actively contributing to the process of ammonium assimilation. So to get a bit deeper, we thought, okay, let's do some single-cell RNA-seq. Can we further refine this picture and see which cell types actually contribute to this process? And what we identified was 12 different cell clusters, cluster 10 being the symbiotic cells. And this is the gene expression of the GS gene, the glutamine synthase, and the GO-GAT genes. So the 2 genes that do the ammonium assimilation in the pathway that I showed before. And what we saw is that specifically GS is active in almost every cell type that we find, irrespective of if it does have symbionts or does not have symbionts. So we asked ourselves, if every cell is contributing to the process, how do they get the nutrients? Because you need that carbohydrates first to fuel that reaction, and you need the ammonium. So we started looking at transporters that could be distributing the ammonium and the carbohydrates throughout all anemone tissues, right? And we found 2 ammonium transporters, AMT1 and RHBG1, and we found 3 glucose transporters that very strongly responded to symbiosis by increasing their expression level. And we found that they were also expressed in many of the different cell types that we had. So we thought, okay, first, can we prove that these transporters are really transporters for what we think they are? The problem being that you assemble a genome and then you annotate the gene by comparing it to sequences from the databases. And only because it has a good match and it looks like it does not necessarily mean that it really has the same specificity. To give you an example, if you look at the genome annotation of 8-GLUT8A, which is the gene that responds the strongest to symbiosis, it is actually annotated as a trehalose transporter. So when we looked at the sequence, it looked like a glucose transporter, but annotation said no. So what we did is we just tested it. So we got ourselves yeast mutants. This is a yeast mutant that does not have any ammonium transporter. And this is a yeast mutant that does not have any glucose transporters. So we took our Aptasia genes for the ammonium transporter and put it in the yeast mutant to see if we can now have the mutant grow in a medium that only contains ammonium as a source for nitrogen. And as you can see, the mutant itself cannot grow. But if we give her the anemone gene, they suddenly can grow under these conditions. And the same is true for the glucose transporter mutants. So the mutant itself, if you only have glucose as a source of carbohydrates, it cannot grow, but with any of the identified Aptasia glucose genes, it can now suddenly grow. So it confirms that yes, these transporters really transport the nutrients that we believe they do. So we then asked, okay, where are they in the tissue? Where are these proteins located? So we got antibodies, and here you see the antibody staining for the glucose transporter in either aposymbiotic anemones or in symbiotic anemones. And the third thing we saw is that in aposymbiotic anemones, these two glucose transporters are not really expressed. We don't really detect any signal. But as soon as the anemone is symbiotic, we now see that the transporter is located in the symbiotic tissue surrounding the symbionts. These are symbiosomes where you have the symbionts isolated with the membrane that surrounds them, and we can see the signal of the transporter being basically present in that membrane, right? And we can see this for all three transporters. Interestingly, GLUT8 is basically expressed everywhere, and most strongly in response to symbiosis. The expression goes up very, very highly. Now, for the ammonium transporter, we saw a slightly different picture. For the RHBG transporter, we saw that in non-symbiotic anemones, it's only expressed at the epidermis, at the periphery, the layer that interacts with the bulk water. While the AMT transporter had kind of a speckled expression, not really clearly localized. However, in symbiotic anemones, these patterns changed. So suddenly the transporter that was only expressed at the periphery of the epidermis, we now found it being expressed in the symbiotic tissue as well as in the non-symbiotic tissue. And when we looked at the symbiosomes, we also found that it is present in the membrane surrounding the symbionts. For the AMT transporter in the symbiotic anemones, we found that it suddenly started to locate exactly where the other transporter was before. So we kind of see a reduction of expression of this transporter in the epidermis, and instead now an expression of the other transporter at the same position. And this was a bit puzzling. Why would you remove one ammonium transporter just to replace them for a different one? It didn't make a lot of sense. So we asked the question if there's maybe a qualitative difference between these two transporters. And one of the things with ammonium transporters is that they can either be unidirectional, meaning they really transport ammonium only in one direction, or they are bidirectional, where they can then transport in either direction depending on the concentrations. So we tested if these transporters are uni or bidirectional by using what we call a methylammonium assay. Methylammonium is toxic. So if you have a transporter that can only transport ammonium into the cell, it will keep pumping methylammonium into the cell until it reaches a toxic level and the cell dies. So we took our yeast mutants and made them transgenic with our Aptasia ammonium transporters and subjected them to 200 millimolar methyl ammonium. And what we saw is that in the AMT1 supplemented mutants, they could not grow in methyl ammonium. So we had only very little growth at the beginning, and then at high concentrations, we did not see any growth. However, for the bidirectional RHBG ammonium transporter, we could see that the mutant could now grow even in the presence of methyl ammonium, which indicated it's a bidirectional transporter, meaning that methyl ammonium does not reach toxic level because you will have an equilibrium between inside and outside the cell. So now we knew that, okay, one transporter is bidirectional while the other one is unidirectional. So we have the bidirectional transporter in the aposymbiotic anemone sitting in the periphery. And an aposymbiotic anemone produces ammonium waste, which has to be excreted. And very likely, this is the role of this transporter. The higher concentration inside leads to an export of the ammonium into the seawater. However, when the anemone becomes symbiotic, this bidirectional transporter is now expressed in the symbiotic tissue where it can transport ammonium in either direction. The unidirectional transporter, however, in the symbiotic anemone is now located in the periphery where it can now import ammonium from outside into the system. And our interpretation is that the moment the anemone becomes symbiotic, it has an excess of these carbohydrate backbones that it can use to assimilate nitrogen, not only internally, but also from the environment whenever available. And this is an important trait because coral reef environments are often very poor in nitrogen. So having the ability to make use of whatever nitrogen is available in the environment, bring it in, and then convert it into amino acid is, of course, a beneficial trait for these organisms. So basically, what we came up with is that we believed that the anemone, when it becomes symbiotic, now uses its entire tissue to try to assimilate as much nitrogen as possible. But this would mean that it would chemically bind the ammonium to the carbon backbones that it gets from the symbionts. And the question was, can we find more evidence to show that this is really the case? So we performed another isotope labeling experiment. And what we did is we took again 13C bicarbonate, which only the symbiont can take and convert into sugar. And we took 15N labeled ammonium. And we said that if this chemical reaction really happens, it means that wherever my carbon is, my nitrogen should be. So if I provide them with both nutrients, I should find a strong spatial correlation between these two signals, right? So we performed nanosims, where you basically have, as you can see here, you visualize the 13C and the 15N isotopes. And you can then overlay the signals and see where they correlate or how strongly they correlate. And what we found was that here we see the correlation of the 13C to 12C ratio and the 15N to 14N ratio. And we have the aposymbiotic epidermis, the gastrodermis of aposymbiotic anemones, the epidermis of symbiotic anemones, and the gastrodermis of symbiotic anemones. And when we look at how well carbon and nitrogen correlate, we do see that in the aposymbiotic individuals, there's hardly any correlation between the signals of carbon and nitrogen. But when we look at the symbiotic anemones, we do find that nitrogen and carbon localization correlates very, very highly. So wherever my carbon is, that's also where my nitrogen is. And interestingly, what we found was that the total amount of the signal that we find in the tissue of the host versus inside the symbionts, because in those images, we can also classify not only the tissue, we can also separate symbiont tissue from host tissue. And then ask how much signal do we find in the host tissues, how much signal do we find in the symbionts? And what we found is that the host tissues together assimilate about similar amounts. It's statistically not significantly different from the amount that the algae assimilate. So host and symbiont assimilate about similar amounts of that nitrogen that I give to the system. So the host does assimilate the nitrogen to control the availability, but of course he cannot control 100% because this would mean that the symbionts do not get any nitrogen at all. So we have a system that basically keeps nitrogen at a level that allows the symbionts to photosynthesize, to be functional, but just not enough to keep proliferating at very high rates. So with this, basically, there was only a few questions left to answer, and this was if this mechanism really controlled symbiont density. We could show before that increasing ammonium increases the symbionts, but according to that model, increasing sugar should actually decrease symbiont density, and we have not tested that yet at this point. The other question is, are we looking at a system that only works in anemones and maybe in corals? Or is this maybe the mechanism that applies to all zooxanthellate cnidarians? So again, the idea behind this system in a fully symbiotic anemone is that the glucose is used to limit nitrogen. So if I increase nitrogen, I should have more symbionts. If I increase the sugar, I should have less symbionts. And we tested this first in our anemone. And here in the plot, you see the symbiont density and below different glucose concentrations. And what we found is that, yes, with increasing glucose concentrations, we see decreasing symbiont concentrations. And as we showed before, if we do the same but with nitrogen, we see that the more ammonium we give to the anemones, the more symbionts we find in the tissues. But this is our anemone, and there are many zooxanthellate cnidarians. And one of the nicest thing about this model is that the only thing it requires is the presence of the molecular machinery to assimilate ammonium into amino acids, and all cnidarians have that. So is this maybe the same mechanism that also allows all of these other cnidarian taxa to engage with Cymbidium, be it jellyfish, octocorals, or naked corals? And here you see an overview of cnidarian taxa, basically, and all classes have species that evolved symbiotic relationships with Cymbidium. Basically, each and every class has at least one species, right? And they evolved them independently. So there must be a simple mechanism that allows for this relationship to just evolve de novo very easily. Specifically, when we look here at the different hosts, because they can also engage with mollusks, foraminifera, even worms, but cnidarians seem to be their absolute favorites. So Cymbidium really love cnidarians. And our thinking was that maybe it is because it is this simple mechanism that allows for these relationships to just pop up very easily. So we thought, why don't we test this in our coral Stolophyra pistillata, again, our anemone Aiptasia, and the upside-down jellyfish, Cassiopeia andromeda. So basically, more than 700 million years of cnidarian evolution, completely independent symbiotic relationship that have been established. So what if we take the coral and we give them ammonium, or we give it glucose? And here again, we have the symbiont density. And what we see is if we give the coral ammonium, as we saw before, the number of symbionts increases. But if we give glucose, the number of symbionts in the tissue drops dramatically. For the anemone, as I shown before, the same, ammonium increases, glucose decreases. But even in the upside-down jellyfish, so a species that is evolutionary, very far away from this, we're talking 700 million years, still the same. Ammonium increases the number of symbionts and glucose decreases it. Now, one of the issues with this is that we know that the stress response of bleaching exists. So the first question somebody would ask is, maybe the reduction in symbionts you see if you give glucose is actually a stress response. Maybe you're just bleaching them, you're stressing them out, and they're kicking out all their symbionts. So we thought, okay, let's do another experiment. We give them glucose and ammonium at the same time, right? So we still have that effect of the glucose, but based on our model, if we also give them ammonium, we should kind of cancel out the glucose that we put into the system. And what we see is this: if we give them glucose, it drops, and if we give them glucose and ammonium, we do not really see a change in symbiont density. And the same is true for our anemone and for our upside down jellyfish. So this kind of showed us that, yes, it seems to be that the same mechanism holds true also in the other cnidarian species. So the last questions we had is this really acting through the GS-GOGAT machinery as we proposed? And are there any differences between the hosts? And this is a rather complex experiment that we've been doing. So we did another round of isotope labeling, but this time we used ultra-high performance liquid chromatography, high resolution mass spectrometry. That's quite a mouthful. Yes, I know. And it's quite complicated. So the awesome thing about this is that we can resolve peaks with a single isotope difference, right? This is the profile only of glutamate. So glutamate should actually have only one peak, which is this 5,2, this peak here. This is the normal glutamate peak that you will have if all carbon atoms in that molecule are 12C and all nitrogen molecules are 14N. But because we give them 13C glucose and 15N ammonium, you now produce all different variations of this molecule, right? The first one being that one nitrogen is actually a nitrogen 15 atom, okay? And this would give you the next peak. So it would shift the peak by 1. The next would be that you have one 13C atom and, yeah, one 13C atom, and everything else is normal 12C and 14N. And that will give you this peak. So basically, your glutamate peak now becomes a number of different peaks that you can all identify and then sum up and calculate exactly how many 13C atoms do I have in my glutamate pool and how many 15N nitrogen do I have in my glutamate pool. So I can now really calculate of the nutrients that I put in, how many of those have been incorporated in my molecule glutamate. And you can do this now with the intermediate metabolites in the pathway that I showed you before. So glutamate, glutamine, alpha-ketoglutarate, O-phosphoryl serine, and glycine. So for each of those molecules, you can now quantify how much of these isotopes are in there. And you can do this for every biological replicate of your coral, your upside down jellyfish, and your symbiotic aptasia and your aposymbiotic aptasia. I would not recommend doing it because there's no software for it. You really need to have a person who analyzes every single peak for every single biological replicate in every single treatment. I think the postdoc still hates me today. I approached him if he would be interested to repeat the study a bit different. He said, no. Well, but if there are people interested, I'm not selling it really well, I think. Anyways, so the first thing I'd like to pay your attention to is that when we look at the symbiotic aptasia, well, no, that was not what I wanted. Yes. So when we look at the symbiotic aptasia on top and we compare it to the profile of the aposymbiotic. So here below is the color code for the different treatments. So control, no isotope labeling, only 15N ammonium, 13C glucose, or both 13C glucose and ammonium, right? And let's just look at the last one. So we give them both ammonium and glucose label. We see that the symbiotic anemone incorporates both the carbon and the nitrogen in glutamate. But we see that the aposymbiotic one does exactly the same thing. It also incorporates it. This is important because it means that it is not the symbiont taking up those nutrients, converting them into glutamate, and then giving the glutamate to the host. It is indeed the host performing this chemical reaction because we see that the apo without any symbionts does exactly the same. There's some differences with regard to the efficiency, and this is because the symbiotic does not only have the glucose that I give it, it also gets the glucose from the symbionts on top of that. So it has a higher capacity to perform this reaction. We also see when we look at how much of this signaler ends up in the amino acid serine that it's about the same amount in symbiotic and aposymbiotic anemones. So again, it doesn't matter if you have the symbiont or not, proving that this reaction is truly host-driven and does not require a symbiont to do it. What we do see, however, is that when you look at the coral, the levels of incorporation of nitrogen are always lower than the anemone or even the upside-down jellyfish. So we can see that, yes, it takes up the nutrients, yes, it converts it into serine and the intermediate metabolites, but at a lesser rate than our anemone and our jellyfish. And the same is true, of course, for glycine here, the same pattern. So the question is, what does the coral do with those nutrients if it doesn't use it to produce amino acids? So here we see the 13C content of the different metabolites, 3-phosphohydroxypyruvate, so the carbon backbone coming from the symbionts, then glutamate, glutamine, serine, and glycine. And what we see is that the coral takes up much more of the 13C, at least we find much more in the carbon backbones. But it does not use it to produce more amino acids. The amount of the amino acids is basically the same as the jellyfish or the anemone. The only ones that take up more of the carbon is the aposymbiotic anemones. So they seem to take up even more of that sugar and use it and burn it. What is also interesting is if we now give these individuals both glucose and ammonium, the picture suddenly changes. So now our upside-down jellyfish and our anemone, who took up very small amounts or much lower amounts of that sugar, take up much, much more and use these carbohydrates to also produce more amino acids. It's kind of like, I give you extra sugar, and the anemone says, yeah, I don't need it because there's no nitrogen for me to scavenge and I have more than enough sugar. However, if I offer her sugar and ammonium, it says, oh yeah, now I can take both and I can use it to produce amino acids. The coral does not do that. The amount of carbon they take up is still the same, and the amount of amino acids they produce is still the same, no matter if I give it glucose or glucose and ammonium. So what happens with this additional sugar that it's taken up that we don't see in the anemone and the jellyfish? And the only explanation we have is that it has a higher metabolic demand to maintain calcification. The calcification process is quite energy demanding because the coral has to keep these proton pumps running all the time and they require ATP. So we believe that this is why the coral takes up more of the sugar but doesn't use it to assimilate nitrogen, but instead burns it to keep the calcification process going. We haven't shown that yet. I'm interested in doing some skeletal analysis to see if I can find any signals of this 13C in the skeleton. So how can we now translate this to anything that has to do with bleaching? Now, the neat thing about this model is that it is not only very simple and can be applied to any cnidarian, and therefore explain why these symbiotic relationships can evolve so easily, but it might also be able to explain why these relationships are so sensitive, because the mechanism that the host uses to control the nitrogen is dependent on what the symbionts provide, the glucose. Without the glucose, the host does not have the carbon backbones to control the pool of nitrogen. So if we end up in a situation like environmental stress that affects the productivity of the symbiont and reduces the amount of glucose the symbiont translocates, it basically takes away the means for the host to control the nitrogen pool. So if the symbionts don't give sugar, the host cannot control the nitrogen availability and therefore symbiont proliferation. And if at higher temperature, you now get less to meet your energetic demands, and you start using up all your reserves, and you now start to burn proteins, you're now even flooding the system with more nitrogen. You burn the proteins because you want that carbon backbone, but as a waste product, you produce nitrogen. And this nitrogen then fuels symbiont proliferation, because the symbiont can say, I have a bit of glucose, I now have nitrogen, I use it to proliferate, because that's what unicellular organisms do, right? And this ends up in a vicious cycle where the symbionts give less and less and less, and basically start taking more and more and more. And it shifts this relationship from mutualism towards parasitism. And we believe that maybe that's the point where the host has to pull the plug, because otherwise the symbiont population would start proliferating at its expense, leach out the nitrogen, and in the worst case scenario, overpopulate within the coral tissues. So in conclusion, what we've been showing is that there seems to be a feedback mechanism that depends on the availability of carbohydrates and nitrogen that kind of regulates this symbiotic relationship. And the host controls this by using carbohydrate backbones to assimilate its own waste nitrogen. The interesting thing is that it's all based on the photosynthesis that the symbiont provides. So the symbionts provide the host with the means to control them. And the interesting thing is that this would mean that this symbiosis is not based on cooperation, but rather on competition. Host and symbiont competing for the nitrogen in the system. We could also show that these mechanisms seems to be universal across zooxanthellate cnidarians, at least in corals, jellyfish, and anemones, and that the corals might require more of the carbohydrates to satisfy energetic demands, meaning that they have less capacity to control nitrogen because they need to use more of the carbohydrates that they get to keep the calcification process running. And this might be because corals are more sensitive to nutrient imbalances compared to, for example, anemones and jellyfish, which can usually withstand higher temperatures than corals can. And yeah, with this, I would like to thank specifically my research scientist, Guoxin Xu, which basically ran all of these studies. And of course, many, many, many collaborators, including Virginia. And many, many others that have contributed to these. And of course, you for staying on and listening to this. Thank you very much. Questions?
And thank you, that was awesome. Tour de force of your work. I was curious about what are the environmental, what's the right stuff in the environment to promote this evolution, this symbiosis? Because I was really struck by the end limitations, because about geochemistry, we think of tropical carbon systems like P limited. That would be you want to constrain somebody, get them hooked, it would be phosphorus, because that's the least limit of the environment. But yet coral uses 10. Is it because it has to be a place where it's a little bit limiting, but you get enough of it? So, is there
very simple answer, right? And that is that we talked about this synergism, right? Is the host a great source of phosphates? No, but it's a great source of nitrogen. Why? Because animals produce an excess amount of nitrogen as waste that we bring to the bathroom. We even have these specialized facilities where we go to just dump it. So you take a photosynthetic organism that is not carbon limited because it can take CO2 and just convert it into sugar, right? But it's limited by other nutrients like nitrogen, for example. And you have the host that is limited by carbohydrates. And you put them together and you create the synergism where the one produces excess carbs that he cannot use and the other produces excess nitrogenous waste that he cannot use. The problem why the host cannot give it freely away is that the symbionts would just keep proliferating and proliferating. So instead, he uses it as a currency to pay, right? This exchange that we meant, that excess carbon that has no real value for the symbionts because they can just produce it out of thin air, literally. And the host with the nitrogenous waste that it would actually throw away, but now, hey, I have a bargaining chip and I can exchange nitrogen for carbs. And you build a system that is built on synergies where both pay less than they actually receive back. right? The question here is, of course, to what extent are we looking at a fair relationship when the symbionts are in a state that we would say is controlled? It's more similar to relationships we have with cows or cattle, right? So we allow them to grow, and then we keep them right there, and we keep milking them. But if we look at the number, at the density of symbionts in the environment, they're much, much lower than the densities that they can reach in the host tissues. So from the perspective of number of individuals in the population, it is still a great deal for the symbionts. And the reason why there is no selective pressure in evolving any mechanisms against becoming a thrall to the host, right? Because it's still beneficial on a population numbers level. And yeah, with phosphate, a lot of colleagues thought that, nah, it's not nitrogen. One of the reasons why it took so long. So nitrogen has been put forward already in the 80s and then the 90s, but the community was very resistant to these suggestions. And as you can see, we tried, and giving them phosphate increases it just a tiny bit. So the main limiting factor is nitrogen. Once you flood them with nitrogen, then very likely phosphate becomes the next one. Yeah.
Okay, very naive, but trying to link up with the bleaching. If you think of the heat stress causing bleaching events, I imagine folks think of the heat shock response and chaperoning functions of the cell as saving the host from bleaching, perhaps, of stress. Is it reasonable to think of this simple machinery as also a source of phosphogenetic variation among host individuals to be able to not tolerate stress, but also manage symbiosis while dealing with stress? Yeah.
So the interesting thing about this mechanism is that it all relies on what the symbiont provides, right? So as long as the symbiont is doing well and produces this excess of glucose or photosynthesis that it can translocate, everything is fine, everything is good. It only breaks down once the symbiont is affected. Only if this goes down, you enter this vicious cycle. Now, what is interesting is that the current theory of what causes bleaching is based on the oxidative stress hypothesis, meaning that increased temperatures impair the photosynthetic machinery, which leads to the production of reactive oxygen species, which then leaks into the host cell and triggers a host cell immune response. Now, under this scenario, ROS is just a symptom of this not working anymore properly, of the symbiont not producing. That's why right now there is no strong evidence showing that ROS itself is really the trigger. Based on this, we would rather assume it might be a symptom that shows us that the symbiont is not doing well. And we already know if the symbiont is not doing well and produces less photosynthesis, it does not say, okay, let's do 50-50. It still says what it needs. So the one paying the price is the host because he's the one getting less, right? The symbiont will first satisfy his own needs and only translocate excess if there is any. And I believe that, yes, from a resilience perspective, working on more resilient symbionts is the better way to go. However, there's a caveat here, right? Because it does not mean that it's completely independent of the host, because this guy is surrounded by a membrane that is surrounded by a host cell, that is surrounded by host tissue. It does itself have no access to the environment. All the nutrients, everything it needs, CO2, oxygen it produces itself, but every other nutrient has to come from the host. So there is very well a host component with regard to how well is the host able to provide to the symbiont. You could take a super resilient symbiont and put it in a host that is not able to provide, and it will just do miserable, because if you don't give him anything, you cannot expect him to perform, right? And this is something where we need to understand to what extent does the host contribute to symbiont resilience. So it's not just an isolated symbiont thing, and you have to look at it from a holobiont perspective. But yeah, I believe if we can make them more resilient.
One last question.
Very interesting. So thinking about sort of the signal to noise ratio at the very beginning, like 11,000 genes in the biological replicates as being differentially expressed, 300 of them were relevant across, and this is the same exact clone, presumably
Exact
it's
clone.
the exogenous same clone.
Same clone.
So,
Yeah.
so do you have any insights of like,
Yes.
what were those genes? And
Yeah. And if you look, that's what I said. If you're interested in these things, look at this paper because we tried to take this apart. When you look at a PCA of all the experiments, they all cluster by source. So we thought we make a PCA and we see all symbiotic anemones on one side and all aposymbiotic on the other side, but that's not what you see. You see experiment 1, experiment 2, experiment 3, experiment. So when you then look at the material and methods and you look at what was the feeding regimen, what was the light intensity, and you start putting on all of these things, and you then ask, how much does each of these factors explain the variance in your experiment? You see that many of those factors actually explain more than symbiosis for this data set. So the experimental conditions have a huge impact. And if you ask why this one experiment had 12,000 differentially expressed genes, I can tell you why. This was an experiment where all symbiotic anemones were sitting in one tub in a light incubator, and all aposymbiotic anemones were sitting in one tub in a dark incubator. So you don't have any batch replication. So anything that affects this batch, it's not only symbiosis, it's how close you are to the light, how close you are to the heater. Everything will influence this. And because you have these stark contrasting environments, you had this huge amount of variation. If we look at our data set that we produced back then, which was I did something. Oh my God, I killed it. Let's see, can I get it back? You want me to quit? Okay, just very quickly. Our experiment we did here, the very first one, 3,000, we had 6 anemones in 3 different batches. And we had the 6 aposymbiotic in 3 different batches in the same incubator, right? So the environmental conditions of the symbiotic and aposymbiotic was the same incubator, and we had 3 independent batches for each treatment to kind of get more variation, meaning that less genes now become significant that affect only one batch.
Well, before we clap for Manny, David Freed has something to say.
So tonight, 5 to 8, night at the museum in Cordley Hall. Join us for a ghoulish display of the collections with lots of things. So this is in the herbarium, the arthropod collection, and in 2316, where vertebrate biology is taught. If you end up in the central courtyard of Cordley Hall, there'll be people who could lead you up to the relevant things.
Thank you, and thank you so much, Manny. That was really